The human stomach operates as an extreme environment. Every day, it produces up to 1,500 milliliters of fluid at a naturally destructive pH of 0.9 to 1.2. This fluid, known as gastric juice, is comprised of 4 distinct biochemical components: hydrochloric acid, pepsinogen, intrinsic factor, and mucus. Understanding how the body safely produces and regulates these components is a high-yield topic for both clinical pharmacology and medical board examinations. Gastric secretion is a precisely calibrated chemical cocktail where specific cellular outputs must be balanced to ensure effective digestion without damaging the stomach's own lining. Anatomically, the stomach is divided into 3 functional zones: the fundus at the top, the central body, and the distal antrum. This diagram shows a gastric oxyntic gland in the stomach's mucosal lining. Notice the deep pits and distinct epithelial cell layers. In the midsection, parietal cells secrete hydrochloric acid and intrinsic factor. Deeper at the base, chief cells produce the inactive enzyme pepsinogen. On this broader diagram, check the distal antrum region. It contains pyloric glands housing distinct G cells. Unlike parietal and chief cells that release products into the lumen, G cells are endocrine. As the bottom arrow indicates, they secrete gastrin directly into the systemic circulation. The stomach relies on this strict cellular and regional compartmentalization to ensure enzymes are delivered to the digestion site, while regulatory hormones are routed through the blood. To understand acid production, we have to look inside a single parietal cell. The cell has 2 critical borders: the apical membrane facing the stomach lumen and the basolateral membrane facing the bloodstream. This diagram outlines acid secretion. Intracellularly, aerobic metabolism produces carbon dioxide. Carbonic anhydrase catalyzes CO2 and water into carbonic acid, rapidly dissociating into hydrogen and bicarbonate. At the apical membrane, an HK-ATPase pump transports hydrogen out, pulling potassium in. Chloride diffuses out to follow. Pushing ions against such an extreme concentration gradient is a primary active process. It requires continuous cellular energy expenditure in the form of ATP. Returning to our parietal cell diagram, look at the blood-facing basolateral membrane on the right. The remaining bicarbonate ion is absorbed into the blood in exchange for incoming chloride. This influx of absorbed bicarbonate creates a measurable spike in gastric venous blood pH after a meal, a phenomenon known as the postprandial alkaline tide. Clinically, this pathway is a major pharmacological target. The drug omeprazole specifically binds to and inhibits the apical HK-ATPase pump. By mapping these exact membrane transport mechanisms, clinicians can safely shut down the acid factory at its source to treat gastric ulcers. This table breaks down systemic regulation. The stomach operates in 3 phases. The first is the cephalic phase, accounting for 30% of secretion. Triggered by the brain via smelling or tasting, the vagus nerve sends signals to parietal and G cells before food arrives. This phase is proven by the experimental concept of sham feeding, where a subject chews and swallows without allowing food to reach the stomach. The vagus nerve alone still triggers robust acid secretion. The second stage is the gastric phase, which drives 60% of total secretion. Looking at the local loops in the diagram, this phase activates when the stomach walls physically distend and when chemical breakdown products like amino acids stimulate the G cells. It is also worth noting that exogenous substances, specifically alcohol and caffeine, artificially stimulate this gastric phase mechanism. The stomach anticipates the meal via neurological hardwiring during the cephalic phase, but it commits the vast majority of its chemical resources only upon physical contact with nutrients. The final 10% of regulation occurs during the intestinal phase, the period when acidic chyme begins exiting the stomach and entering the duodenum. The primary purpose of this phase is actually inhibitory. When the intestinal mucosa experiences distension or chemical irritation, the enterogastric reflex halts further stomach activity. As listed here at the bottom of the page, the presence of acidic or fatty chyme triggers the release of specific GI hormones. Secretin, cholecystokinin, GIP, VIP, and somatostatin all act to hit the chemical brakes on gastric secretion. Without these rapid neural and hormonal cutoffs, the highly acidic chyme would quickly ulcerate the delicate duodenal lining. Returning to the components of gastric juice, the primary digestive function is clear. Hydrochloric acid provides the highly acidic medium required to convert inactive pepsinogen into the active protein-cleaving enzyme pepsin. However, the most vital role of the stomach is its hemopoietic function. The secretion of intrinsic factor by the parietal cells is the only stomach function absolutely essential for human life. Intrinsic factor is required for the terminal ileum to absorb vitamin B12. Vitamin B12, in turn, is strictly required for the successful maturation of red blood cells in the bone marrow. Without parietal cells producing intrinsic factor, such as after a surgical gastrectomy, a patient loses the ability to absorb B12 and will develop pernicious anemia. The stomach acts as a central hub for metabolic health, linking chemical digestion directly to the production and maturation of red blood cells. Let's run a rapid drill to lock in these high-yield facts. Match the cell to the secretion. Parietal cells secrete hydrochloric acid and intrinsic factor. Chief cells secrete pepsinogen. Remember the exception. G cells are endocrine. They secrete gastrin into the bloodstream to stimulate parietal cells indirectly. For regulation, the gastric phase drives 60% of total acid secretion, triggered directly by stomach distension and the presence of amino acids. You now possess the mechanistic foundation to confidently break down the physiology of the gastric engine on any board or essay examination.